So here we are going to look at DNA replication today. Um, this is kind of a complicated process that has a lot of words in it, and so we're going to use this lecture as like a prequel to class on Thursday, Friday, so that you can come to class ready to talk about this and learn all about it. So what I want you to do today is you're going to have a cheat sheet. So have a separate sheet handy from your notes that you can always pull out and look at, because we're going to talk about a lot of different enzymes and a lot of different terms today. And as we learn these new terms, we can summarize them on this sheet. And so if you're looking like what terms do I write down, you're going to see them in red today in our PowerPoint. Okay, so facts about replication. DNA has to be copied before the cell divides. DNA is copied during the S or synthesis phase of interphase, and new cells will need identical DNA strands from the original cell. So remember things in blue. Let's not write this down because you guys already know all these things. I know you do because we spent a lot of time talking about this in mitosis. But you need to remember that your new cells are going to be identical to your mother cells. Your daughter cells are identical to your mother cells. So that synthesis phase, the S phase during the interphase of the cell cycle, remember blue things we're not writing down. It happens in the nucleus of eukaryotes. Remember eukaryotes are those with nuclei. Prokaryotes are things like bacteria and archaea. They don't have the nucleus. And DNA replication takes place during the S phase, which happens between G1 phase, G2 phase, before mitosis. Mitosis is where it all splits up. Okay, DNA replication. So this begins at the origins of replication. Okay, this is a word you need to know, origins of replication. Basically, it's where replication starts. So take out your cheat sheet, write down origins of replication on it, write down the word, write down a summary of where DNA replication starts. So replication meaning copying, creating a new identical DNA strand. And they open, the two strands, they open up and they form replication forks, which is a Y-shaped region where replication occurs. And these new strands grow at these forks. So we have a DNA strand here, kind of looks like a ladder, and it's going to split at the replication fork, and new DNA strands are going to grow from here and here. DNA replication, as the two strands begin, they open at the origin and form replication bubbles. So another red word, write that down on your cheat sheet. And eukaryotic chromosomes have many replication bubbles. Looks something like this. So your DNA, it's untwisting and forming little bubbles where DNA is replicating and making new strands that are identical to the original copy. Prokaryotes, on the other hand, those bacteria, they just have a single replication bubble. And they only have a single replication bubble because there is just one single chromosome in that cell. And so they have two replication forks instead of many replication forks. So originally, we had the blue DNA. And that blue DNA is being replicated and filled in with this green DNA. So you can see half of this chromosome is copied DNA, and the other half is still the original DNA, and we'll end up eventually with two identical circular chromosomes for this bacterial cell. Alright, DNA replication continued. So the enzyme helicase, red word there, helicase unwinds and separates the two DNA strands by breaking the weak hydrogen bonds. So helicase, anything that ends in the word ASE is an enzyme. Enzymes are really important for making things happen in your cell. They're proteins, and they are the action makers in your cell. They make sure that things can happen. So helicase is an unwinding enzyme. The single strand binding proteins attach and keep the two DNA strands separated and untwisted. So there are proteins that are attached, and they just make sure that those two DNA strands stay separated. Alright, DNA replication. The next enzyme that we need is topoisomerase. So that's kind of a big word there, but it's like two words, topo or topo, isomerase, and it attaches to the two forks of the bubble to relieve stress on the DNA molecule as it separates. So imagine you're untwisting, let's draw a picture of this. Imagine that you have your helix here, right? It looks like this, and it's twisted up. But as it becomes untwisted, all of the loops down there get shoved to the bottom, and that becomes twisted really tightly. Right? And all of that stress, there's a lot of stress on the DNA right here. And so we don't want that to like damage our DNA, and so this topoisomerase comes in and relieves stress at these points right here, at all these tense sections of the DNA of the replication bubble. Oops, apparently there's more. All right, DNA replication. Before new DNA strands can form, there must be RNA primers present to start the addition of the new nucleotides. So RNA primers basically make new sections of DNA. They're like the new nucleotides being added. And primase is the enzyme that creates RNA primer. So RNA primers are basically the thing that creates new nucleotides. Primase is the thing that makes the things that make new nucleotides. So primase makes RNA primers make new nucleotides. DNA polymerase is then responsible for attaching these new nucleotides onto the DNA so that we can have a whole, complete strand. So all these words in red, make sure they're written down on your cheat sheet. If you're having a hard time like summarizing them, you can check out like online. I'm gonna look for some like tutorials on this too to help show you guys, to help you better understand this. Okay, so step one, the helicases unwind the DNA, and then we have a replication fork here. The complementary nucleotides are added to each strand by the DNA polymerases, and then the two DNA molecules that form are identical to the original DNA. So this strand here is identical to these two new strands. So we have two strands being added to, like, side by side, identical to each other. All right, DNA replication again. So DNA polymerase, remember that's what adds, like attaches the DNA nucleotides to the DNA. It can only add nucleotides to the 3' end of the DNA. So you should have a 3' like summary in your cheat sheet there. Basically remember that the glucose molecules, they have 5, each of their carbons are numbered, 1, 2, 3, 4, and 5. And sometimes the 3 faces up, sometimes the 5 faces up. But the nucleotides can only be added to the 3' side. So our direction of replication goes from 5' to 3'. So our RNA primer must go down first, and then DNA polymerase adds on from there. So new strands are built in a 5' to 3' direction. So maybe just write that sentence down in your cheat sheet. That's really important to know. New strand built in a 5' to 3' direction. Okay, so let's review. How are those carbons numbered? So 1, 2, 3, 4, 5. And our nitrogen bases are added on the 3' end. And remember that these strands are anti-parallel. So one goes from 5' to 3', while the other side goes from 3' to 5'. So the leading strand, we have to talk about how these new DNA strands are created. So the leading strand is synthesized as a single strand from the point of origin toward the opening of the replication fork. So this is a picture of our leading strand. It is one single strand. It's the like starting strand, and it goes from 3' to 5'. So the leading strand adds on from 5' to 3', and then the 3' to 5' here. All right, new DNA strands. There's also something called a lagging strand. It is synthesized, so created discontinuously, so not in a like continuous manner against the overall direction of replication. So the lagging strand is made from many short segments, and it's replicated from the replication fork towards the origin. So it goes from, here's your leading strand. Your lagging strand is synthesized in the opposite direction and synthesized in fragments, in little pieces, piece by piece. While the leading strand goes one by one by one in a row, lagging strand is created in segments. Okay, so the short segments that are created on the lagging strand, we call those Okazaki fragments. So I would put that in your notes there. And they have to be joined together by an enzyme. And we call that enzyme ligase. So ligase brings those Okazaki fragments together to make one strand. So we had an Okazaki fragment here, Okazaki fragment here. We bring it together with one enzyme called ligase. So our leading strand is created 3' to 5' continuously, while our lagging strand goes from 3' to 5' again, but in segments. Okay, so we're gonna, it's all in blue here, we're not gonna write this down, but DNA polymerase, initially it makes about 1 in 10,000 base pair errors, and then the enzymes go through and they proofread and correct these mistakes. And so our new error rate for DNA that has been proofread is about 1 in 1 billion base pairing errors. We call we call this a semi-conservative model of replication. Basically, we are making complementary strands of DNA. So as those molecules separate, it's like a template, and we make new complementary strands. They're identical, but there's the opportunity for errors. So we call it a semi-conservative model of replication. But new DNA is half parental DNA, half new DNA. So you always keep part of what you had before. There are things that can damage our DNA, like chemicals and ultraviolet radiation. So we always have to repair this. And sometimes we take out lots of different parts of DNA. Alright, so think about this strand of DNA and look for the complementary sequence. Let's see what the complementary sequence would be. And I'm going to end this lecture here because it's about time for class to start. Alright.